In this video, we will discuss about a different idea where they use this adipocyte to suppress the tumor progression. The idea is that the adipocytes, the engineered adipocytes, will take up the nutrients and thereby leave the cancer cells with lack of nutrients, or cancer cells will be deprived of the nutrients, and that will cause the tumor suppression of this tumor progression. So this is the schematic of the experiments that they did. Is that they take this CRISPR AAV adipocytes, and they used some of the genes that are responsible for this increased nutrients consumptions, and they co-culture them in this transwell plate where the this CRISPR A adipocytes are on the top side of the transwell, and cancer cells are on the bottom side of the transwell. And what they are able to see, and they used 5 different kind of cancer cells, breast cancer, and 2 different kind of breast cancers, colon cancer, pancreatic cancer, prostate cancer. And for the negative control, they used this CRISPR AAV adipocytes with the gene that does not cause high fatty acid consumptions. So when they check in the microscope, they are able to see that compared to the control, in the other genes or adipocytes with other genes has lower number of cells compared to the controls, and this conditions is true for all the 5 cancer cell lines. Next, they checked and this is the quantifications. In the panel C is the quantifications, where we can see that compared to the control, for all the 3 genes that they checked in the adipocytes, they are able to see significantly reductions in the number of cells, indicating that the adipocytes somehow suppress the cell numbers. And the hypothesis behind this is that these adipocytes, which has the high nutrients uptake because of these genes that are transfected, or genes that are transduced, such as UCP1, PPARG, C1A, PRDM16. They cause high consumptions of the nutrients by the adipocytes, and the tumor cells will get deprived of the nutrients, and this could be the reasons why number of cells is low compared to the control in all the 3 genes. Thereafter, they check the proliferation marker, that is Mi67. So this proliferation marker also goes down compared to the control in all the 3 cases, except for this PANC1 and DU145. For this P and this PRDM16, the decrease is not statistically significant. Next, they check the basal glycolysis and maximum glycolysis using this ECAR. That is, I'm not able to recall the full form. It's something related to external acidification, extracellular acidification rate, something like this. And because using that, they check the basal glycolysis and maximum glycolysis, and they are able to see that it is also statistically lower in most of the cases compared to the control for both basal glycolysis and maximum glycolysis. Then they also check the glucose uptake. By in using this luminescence and insulin stimulated glucose uptake. This is the normal glucose uptake, and this is the insulin stimulated glucose uptake. And in both the cases, they are able to see compared to the control. In the other cases where cancer cells are co-cultured with adipocytes using this genes, shows a decreased glucose uptake, and for both the basal glucose uptake and insulin stimulated glucose uptake compared to the control case. Then they check some of the markers that takes place in this glucose consumptions, that is GLUT4 and GCK, and those are also goes down, and the results are similar to what they have seen for the previous cases, that is basal glucose uptake and insulin stimulated glucose uptake. That is GLUT4 and GCK also goes down when the cancer cells are co-cultured with UCP1, PAR, GC1A, PRDM16 genes. The transduce in the adipocytes, and those adipocytes when co-culture with these pancreatic cancer cells, this cause a decrease in the GCK and GLUT4, which is the markers for these glucose consumptions. Next they check this OCR using the palmitate treatment. Palmitate contained media means that they transduce, or they keep this high fatty acid media into the, they keep this high fatty acid palmitate into the cell culture media and then check its consumptions, and they are able to see that its consumptions is lower in these cases when the cancer cells are co-cultured with adipocytes that has these genes, that UCP1, PPAR, GC1A, PRDM16, and in the control case it shows the high OCR levels, indicating that the consumption rates are high for these adipocytes and they are lower for the cancer cells, as most of the nutrients are being taken up by adipocytes. Then they check the other markers, that is CD16 and CPT1B, and its levels is also related, and this CD36 and CPT1B is related to the mitochondria functions. So this indicates the mitochondria functions is also lower in the cancer cells when the cancer cell co-cultured with the adipocytes with these genes transduce. And this, in this case, the results are not uniform for all the markers, all the cancer cells, but for some of the cancer cells, such as for CD36, the results are significant for this MDA, MB436, and for this CPT1B, they are significant for this MCF7, and also for this SW1417, except for the PPAR GC1A. So this overall, this whole results that are presented in this figure points towards that adipocytes somehow can be engineered to suppress tumor growth progression by depriving the tumor cells of the nutrients that are necessary for its survival and progressions. So that's all for this video. If you need to understand more about these experiments in detail, you can go through the reference that has been provided in the description. That's all for this video. Thank you for your time.